Okay, so before we start the spinal cord, there's just some couple of slides that we need to talk about. One of them we have talked about on the previous lesson. We need to discuss the blood-brain barrier because it's a very important exam question that you're going to have. So first of all, do you remember which cells participate in the blood-brain barrier?
Astrocytes.
Very good. We have two types of astrocytes. We have protoplasmic and fibrous astrocytes. What is the location of the protoplasmic astrocytes? They are in the gray matter, while fibrous astrocytes are going to be in the white matter. So if you look at the brain as a box, the outer part of the brain is which matter, gray or white?
Gray.
Okay, so this is where the gray matter would be. So this is the location of the protoplasmic astrocytes. The inner is the white matter. That's where the fibrous astrocytes are going to be. Both of these astrocytes participate in the blood-brain barrier. As you remember, astrocytes, they have lots and lots of food processes, and these feed processes are touching two things. If here we have a neuron with the axon, cell body, and dendrites, and here we have a blood vessel, in between these two structures, you will always find an astrocyte. Okay, so that's where the astrocyte will be, between the neuron and a blood vessel. Food processes of the astrocyte are going to be located on the surface of the blood vessel, specifically where two endothelial cells are going to have a junction. So a food process of the astrocyte always ends up in a place where the two endothelial cells are meeting. Between these two endothelial cells, there's going to be a tight junction. Can you remind me which proteins are present in a tight junction between two endothelial cells?
Claudin, occludin.
Very good, very good. So occludin, claudin, and what is the last one?
Jam.
Very good, jam. Occludin, claudin, and jam. Jam is a junctional adhesion molecule, is not present everywhere. It's only present between endothelial cells specifically. So for example, you have a tight junction between two epithelial cells. Here you will find occludin and claudin. But tight junction between two endothelial cells will possess occludin, claudin, and jam. So why are they important? Because in the exam, you will see a sentence like that. The food processes of the astrocytes induce formation of occludin, claudin, and jam between the two endothelial cells. That sentence is correct. The food processes of the astrocytes are inducing the formation of a tight junction. You just have to remember, what does a tight junction consist of, right? And tight junction is going to consist of occludin, claudin, and jam. So basically, without astrocytes, the two endothelial cells will not be having a tight junction between each other. Okay, so without astrocytes, it will not be there. So what is a blood-brain barrier? That's the question. Very easy. It's simply a tight junction. That's it. So there aren't any layers. Remember, like blood-thymus barrier last semester, it was 6 layers. Blood-air barrier, it was 4 layers. Blood-brain barrier is simply a tight junction between two endothelial cells. That's it. Is it impermeable? Sorry, is it impermeable? It is quite permeable. It allows only certain things to pass. Okay, so typically these certain things are going to be gases. Okay, so gases can easily squeeze in back and forth. But certain things like glucose, amino acids, they need a transporter. So they can't simply just diffuse. You will need a certain transporter. Could you remind me which GLUT transporter is in the brain?
GLUT3?
Nope.
1.
GLUT1. And where else is GLUT1 located?
adipose tissue.
RBCs, brain and red blood cells. Okay, so this is the place for the GLUT1 transporter. So yeah. There's some areas where we do not have blood-brain barrier. Okay, so it's not present there. The endothelial cells in these places do not possess the tight junctions. So we call these organs circumventricular organs. So these organs that you can see here, they are called circumventricular. They're called circumventricular because they are located around fourth and third ventricles in the brain. So that's why you call them circumventricular. They're around the ventricles, right? So circumventricular organs are organs that do not possess the tight junctions between endothelial cells. What are these organs? Pineal body. Quickly, what does it make?
Melatonin.
Very good. It makes melatonin. Posterior lobe of the hypophysis. What does it make?
ADH.
ADH and oxytocin.
I caught you because it doesn't make ADH and oxytocin.
It stores the
It
hormones.
stores ADH and oxytocin. So posterior lobe of the hypophysis does not make anything. It simply stores ADH and oxytocin. Who makes them?
The anterior lobe, the hypothalamus.
Hypothalamus, but say better.
The magnocellular nuclei.
Perfect. Magnocellular nuclei make the ADH and oxytocin, the supraoptic paraventricular nuclei, you remember, of the hypothalamus. They're responsible for that. Median eminence, do you remember what is that?
It was a connection between the
Infundibulum and the third ventricle.
Yes, very good. Between infundibulum and hypothalamus. Okay, so infundibulum, hypothalamus, and then you have here the posterior lobe. So between the infundibulum and hypothalamus, there's a bit of tissue here, which you call median eminence. So yeah. And lastly, area postrema. You will talk about this area a lot in neurophysiology because area postrema is considered to be the vomiting center of our body. Okay, so this is the vomiting center. Area postrema is located in the medulla oblongata. Okay, so this is the area where it is located, medulla oblongata. It is a vomiting center, so anything can make you vomit, anything. Sometimes too much blood rushes to this area, it stimulates area postrema, you will vomit. Certain food molecules can actually dissolve through the blood because there is no BBB, so they can dissolve, irritate area postrema, you will vomit. In neurophysio, you'll talk more details about it. For neurohisto neuroanatomy, most important is that it is vomiting center. It is located in the medulla oblongata, and it's the area where you don't have any BBB. This is additional question that they always ask. We have to learn the difference between external glial limiting membrane and internal glial limiting membrane. They are not the same thing. You remember glia limitans from last class? What was the location of glia limitans?
In interior to the pia mater.
Very good. It was under the pia mater. So it was right under the pia mater. And what was the function of glia limitans?
The movement of neuron or the process of neuron is limited.
Exactly. It was limiting the location, the movement of the brain neurons, because brain neurons, they love to go different places, right? So this particular membrane was making sure that that does not happen. Could you remind me who made the glia limitans? What does glia limitans consist of? So think it is right under the pia mater. So we have brain, this is the pia mater, and between the brain and pia mater is the glia limitans.
Pia mater.
Pia mater is this one, it is a separate layer by itself. Who made the glia limitans?
Protoplasmic.
Perfect. Food processes of the protoplasmic astrocytes. You remember they were coming out and these food processes were creating this glia limitants. Another name for glial limitants, it is also known as the external glial limiting membrane. Okay, so glial limitants equals to the external glial limiting membrane. Internal glial limiting membrane has another name. It is also known as the perivascular limiting membrane. Okay, so it is also known as the perivascular. So it is around the it is around the blood vessel. So what does it participate in? It participates in creating the BBB. So any astrocyte can make internal glial limiting membrane. Protoplasmic can do it, right? Fibrous can do it. But external one is only created by the protoplasmic because that's the most outer one. Does it make sense? Okay. Let's take a look. So neuroglia, chial gold impregnation. This is a piece of your brain. In this piece of the brain, you can see that there is the cortex, so gray matter, and there is the medulla, which is the white matter. Okay, so cortex and medulla. The purpose of the slide is to look at the neuroglia. Of course, we will not be able to see all of the neuroglia. The only cells that we will be able to see are astrocytes. Okay, so here we're talking about which cells you can see. We're only going to be able to see astrocytes. So first of all, these things right here, this is a blood vessel. Okay, so these lines that you can see here, these are blood vessels in the brain tissue. What are these cells?
Astrocytes.
They are astrocytes. What we should ask ourselves, are they protoplasmic or are they fibrous? What do you think?
Protoplasmic.
Absolutely correct. That's protoplasmic astrocytes. How do you know that they're protoplasmic?
Because they're branching.
Very good. The processes are short. Do you see? They're relatively short. And there's lots of branching happening. Because these are protoplasmic astrocytes, this slide is from the cortex or medulla of the brain.
Cortex.
Cortex, because cortex is the gray matter and protoplasmic astrocytes are located in the gray matter. Take a look here. So these are still blood vessels. You can see lots of astrocytes are literally sending their feed processes, right? You can see it clearly on the picture here that they're really trying, right? So you can see this part right here. How would you call it?
Internal glial limitant.
Very good. This is the internal glial limiting membrane or perivascular limiting membrane, right? So that's just a bunch of food processes of the astrocytes, which are helping to create the BBB, right? So it's right here. You can see it on the blood vessel. But take a look at these astrocytes. Do they look the same as before? No, you can see that the food processes are much longer and they don't branch as much. So which astrocytes are these? Fibrous astrocytes. That means that this is the medulla of the brain, or in other words, white matter, right? This is the white matter of the brain. So from these pictures alone, you must be able to say, right? So this is a protoplasmic, or is this the fibrous astrocyte? Okay, let's begin the spinal cord. We cannot talk about neurohistology of the spinal cord without actually knowing a bit of the anatomy. So this is a cross section of the spinal cord. In the spinal cord, the matters are the opposite, right? So if in the brain, the outer is gray, the inner is white, in the spinal cord, it's the opposite. In the embryo, we will learn why is that. So all of the white matter is outside and the gray matter is inside. The gray matter in the spinal cord is organized into horns. We have the ventral or anterior horn, and we have the dorsal or posterior horn, right? You can see that it has this particular butterfly appearance of the gray matter. Surrounding the gray matter, we have the white matter. And white matter is organized into funiculi. So particular organization of the white matter, we call it funiculus. There are 3 funiculi. there is the posterior funiculus, there is the lateral funiculus, and there is the anterior funiculus. So could we quickly specify what would you find in the funiculus? What does funiculus consist of?
White matter.
Axon.
But yes, be a bit more specific. So what exactly is going to be there? Is going to be a bunch of axons. Okay, so funiculi, they consist of a bunch of axons. What about gray matter? What would you find there?
Cell bodies.
Very good. Cell bodies. Okay, so you will find a bunch of cell bodies here. How do you know that this is the anterior and this is the posterior side of the spinal cord? You have to look for the arteries, because you will see that there is going to be only one anterior spinal artery, and there are going to be two posterior spinal arteries. There are going to be two of them on the sides. So in your slide, always try to find a blood vessel. If there's only one in the center, that's the anterior side. If there are two at the back, that's the posterior, right? So one anterior spinal artery and two posterior spinal arteries. Another thing that will help you to kind of know that you're looking at the anterior or posterior side is whether you can find the sulcus or the fissure. Could you quickly tell me what's the difference between sulcus and fissure?
Sulcus is like a groove, while as fissure is like sort of an opening.
Very good. In other words, fissure is much deeper, right? It's more deep, it's more elongated, and etc. Sulcus is very superficial, yeah? So it's just a little bit, that's it. Fissure is quite deep. It's, as you said, it's an opening almost. So there is anterior median fissure, and there is posterior median sulcus. So try to look at the two sides. If you see that one of the openings is a bit more deeper, right, and it is more open, if I may, right? So this is definitely going to be a fissure, and fissure is anteriorly located. So it has to be the anterior side. Okay, so generally speaking, your spinal cord is divided into 31 segments. We have talked about it previously, right? You remember 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, 1 coccygeal segment. And from these segments, we have roots kind of coming out. We have the ventral root and the dorsal root. The two roots unite, creating a spinal nerve. Spinal nerve continues as the peripheral nerve, right? So that's kind of a general distribution of the tissue here. Butterfly we have talked about, they always talk about it, right? So there's a butterfly shape gray matter, and this is a very good picture that you can see, right? So this is a fissure, or is it a sulcus?
Fissure.
Fissure, okay, so you can see it's very deep, very open, so this is a fissure. On the surface, this little tiny thing is the sulcus, that's it, okay, so this is the sulcus. Your spinal cord is going to be covered by meninges, the same as your brain, because spinal cord is also part of the central nervous system, so it's also going to be covered by meninges, the dura, arachnoid, and the pia mater. Inside of the spinal cord, you can find a canal. This canal is known as the central canal. Central canal is the place where cerebrospinal fluid will be present. Cerebrospinal fluid flows into the central canal from medulla oblongata, because you remember, right? Medulla continues as spinal cord. Which ventricle is in front of the medulla oblongata? This is going to be fourth ventricle. So cerebrospinal fluid flows from the fourth ventricle in front of the medulla into the central canal of the spinal cord. Where is the central canal located? It is located in this little bridge-like structure which connects the two sides of the gray matter. This bridge is called gray commissure. Okay, so this area right here, this is known as the gray commissure. It's simply the area which sort of connects the two things. Okay, white matter is going to consist of only myelinated and unmyelinated axons. That's the only thing that you can find in the white matter. That's it, only axons. But in the gray matter, you can find everything. You can find the cell bodies, you can find their dendrites, you can find some axons and even neuroglial cells. Okay, so remember the major difference between them. So white matter, only axons. These axons could be myelinated or unmyelinated, but in the gray matter, you can find anything, cell bodies, dendrites, and axons. How do we call a bunch of functionally related cell bodies together? Here.
Motor
Nucleus. Very good. We call it nucleus. Ganglion is in the peripheral nervous system, but spinal cord is the central nervous system, so you will call it a nucleus. So where the nuclei are located in the spinal cord? In the gray or in the white matter?
Gray.
In the gray, because that's where the cell bodies are. How do we call functionally related axons?
Nerve.
Tract.
In the peripheral nervous system, you will call it a nerve, for sure. In the central nervous system, you could call it so many things, but the one that we call in the spinal cord is actually a tract. So tracts are going to be present within the white matter of the spinal cord. The tracts are, I repeat, functionally related axons, right? So they're related to each other. They have similar functions. These tracts could be going to the brain and from the brain. It's a bi-directional pathway, okay? So within the white matter, you can see that they're going back and forth. Okay, so we have talked about this. This is just a repetition. I'll talk about it in a sec. This is an exam question. They're going to ask you the layers. Okay, so vertebral bone epidural space is above the dura mater, subdural space, right? Then arachnoid, subarachnoid space, that's the place where the cerebrospinal fluid will be, and then the pia mater. Okay, there are certain things which are only present in the spinal cord and they aren't present in the brain. These things are called denticulate ligaments. So we need to talk a little bit about them. So you can see that this is your spinal cord, right? So there are lots and lots of tissues here. You can see that this is a sulcus. It's not very deep. You can see that there are lots and lots of nerves, right? So kind of coming out there. And you can see that it is covered by some sort of meninges. But there's some kind of triangular tissues which are attaching your spinal cord to arachnoid matter. Okay, so these denticulate ligaments, they are pia that is attaching to arachnoid. Yes, so if you draw it in the brain it's just dura, arachnoid, and pia. That's it. But if you draw the same idea in the spinal cord, there's dura, there's arachnoid, there's pia, but pia has a triangular connection to the arachnoid. And this triangular connection is called denticulate ligament. Denticulate because it looks like a tooth. If your tooth are triangular, okay, so it looks like a tooth. So what do you think? What could be the job of denticulate ligaments? Why is it important that we have them in the spinal cord, but we don't have them in the brain? Why these triangular connections are important?
Stabilize the spinal cord.
Perfect. One of the functions is for stabilization of the spinal cord. These denticulate ligaments sort of keep your spinal cord in place, so it doesn't move much. But there is a second function that short Zoli loves. Okay, so What is here? If this is dura, this is arachnoid, this is pia. What is in this space?
CSF.
CSF.
Blood vessel.
Yes, CSF. Blood vessels are going to be there as well, but CSF is what is important, okay? So cerebrospinal fluid. If we didn't have denticulate ligaments, what would happen to all of the CSF?
It will flow out from. Like
It will, due to gravity, all of the CSF will be in your sacral region. That's where it will be. All of this fluid, due to gravity, will be brought up to the bottom, right there. We don't want that. So this is why there are 21 pairs of denticulate ligaments, and you can think about them as levels, okay? So some kind of levels that CSF flows through. It's like additional floors that making sure that all of the CSF is not just flowing down due to gravity, okay? So it's a little bit like it provides a resistance for the flow of the CSF, right? So there is no turbulence that all of it just due to gravity goes down like that. Does it make sense? So it divides the subarachnoid space, so CSF can flow slower. So it doesn't just all go down due to gravity. Okay, arteries, we have talked about them. Dorsal root, ventral root, you remember, right? So dorsal root is the one which is coming in, ventral root is the one which is going out from the spinal cord. In the dorsal root, we have something. What is there? What is in the dorsal root? Dorsal root ganglion. What is located in the dorsal root ganglion?
Cell body.
Cell bodies. Whose cell bodies?
sensory.
Sensory neurons. Which sensory neurons specifically? The somato and the viscerosensory. Both of them are going to be here. Somato and viscerosensory. Which kind of neurons are they?
Pseudo-unipolar.
Very good. They're pseudo-unipolar. So the cell body is here. A little bit of axon is also going to be here. But the rest of the axon is in the periphery, right? It is in the dorsal root and it is in the spinal nerve. That's where the rest of the axon is. So a question, is there a synapse in the dorsal root ganglion?
No.
No. Where do sensory neurons synapse?
Dorsal horn.
Perfect. They synapse in the dorsal horn. Okay, another name for dorsal horn, this is also known as the posterior horn. What exactly do they synapse with? What can sensory neurons synapse with in the dorsal horn?
Interneuron
And
motor neuron.
Which motor neuron? Somatic or autonomic?
Somatic.
Somatic.
Autonomic. Okay, so in the dorsal horn, these sensory neurons could synapse with two things, with interneuron or with the autonomic motor. The autonomic motor is the one which you don't control whatsoever, right? Interneuron, in turn, can synapse with somatic motor. So you're right to say that sensory neurons can synapse with somatic motor, but they do not synapse with them directly. They use interneuron to do so. So how do we call it? The sensory neuron synapses with interneuron, which in turn synapses with somatic motor. How do we call it?
Reflex.
Reflex. Very good. We call it a reflex. A very good example is like patellar reflex, right? When I hit your knee, that's the sensory neuron, right? Connects to the interneuron, connects to the somatic motor. Somatic motor innervates my skeletal muscles of the leg, creating this jerky movement every time you hit the knee, right? Okay, Okay. I feel like I want to talk about something here. Just a second, guys. So we talked about dorsal root ganglion. Yes. We talked about the interneurons. Okay, then let's talk about this way. Yes. So you remember we have functional neurons. We have the sensory neurons, we have the interneurons, and we have the motor neurons. Quickly tell me, where are the cell bodies of sensory neurons?
In the dorsal root ganglion.
In the dorsal root ganglion. Where are the cell bodies of interneurons?
In the gray matter.
Be more specific.
Dorsal horn.
In the dorsal horn of the gray matter. Where are the motor neurons?
Anterior.
Ventral horn.
In the ventral or anterior horn of the gray matter. So we're talking about spinal cord, and lots of students make this mistake because another way to describe anterior horn is to say that this is a motor horn. Okay, anterior horn of the spinal cord is a motor horn because it contains motor neurons. So lots of students, they make a mistake assuming that the posterior is the sensory, but it isn't. Okay, posterior, it's not the sensory, because what do we have here?
Synapse.
We have the synapse of sensory with
Interneuron.
interneurons. The posterior horn or dorsal horn is the place for the interneurons. Does that make sense? The sensory neurons are actually in the ganglion. That's where they are, the cell bodies at least. Okay, guys. There is something which is called coma tract of Schulze. Coma tract of Schulze is a triangularly shaped tract. So what does it consist of? It consists of a bunch of axons, and these axons are less myelinated. Be careful, they aren't unmyelinated. They simply are less myelinated. So as a result, this tract looks eosinophilic. Okay, so it looks very eosinophilic. Where is coma tract of Schulze? It's here. Okay, so we will see it exactly like this. It's a triangularly shaped red area. The function of coma tract of Schulze, we will talk about it in a second. Okay, so just I want to introduce it right now. Okay, there are some kind of enlargements that are present in the spinal cord. There is a cervical and lumbar enlargement. You need to remember their exact locations for neuroanatomy, right? So cervical enlargement is approximately where the C4 to T1, and lumbar is approximately where T11 to L1. So funiculus, you remember, that's organization of the white matter. We have the posterior funiculus, we have a lateral funiculus, and we have anterior funiculus. You need to remember that in the posterior funiculus, you will only find ascending tracts. So in this posterior funiculus, the axons are going to where? If they're ascending?
To the brain.
To the brain, okay? They're going to the brain. In the posterior funiculus, there are tracts, axons, and they're going to the brain. So what do you think? Which kind of information are they carrying?
Sensory.
Sensory, very good. They must be sensory because they're going to the brain. The brain has to understand, like, what are you touching, what is going on. So in the posterior funiculus, I repeat, there are a bunch of ascending tracts. Ascending tracts are sensory. In the anterior funiculus, you will find mostly descending. Mostly, not only, mostly descending. Descending are going from the brain to the spinal cord. That means that they are not sensory, they are motor. They're motor. In the lateral funiculus, you will find both. They're mixing. Okay, so there's a certain tract which is, for example, bidirectional. It can go up the brain, and the same tract is used to send information down from the brain. So bidirectional tracts are going to be present in the lateral funiculus. Is it clear? I should have mentioned that today's lesson is the most difficult out of all of the lessons, by the way. Okay, so I should have mentioned that. So there's lots and lots of information that is very much highly tested, both neuroanatomy and neurohisto. There's additional tract, which is very special. It's called Lissauer's tract. Another name for Lissauer's tract, it is also known as posterolateral tract. Lissauer's tract is here. It's this area. Okay, so this is where Lissauer's tract is present, posterolateral. You can see on this picture, they're saying fasciculus. Fasciculus is another way of saying tract. You remember our first lesson, right? So tract, fasciculus is identical. So you can say comma fasciculus of Schulze, because tract is fasciculus, right? So a question for you guys. What do you think is present in the Lissauer's tract?
Axons.
Very good. Whose axons? That's the question. Whose axons are there? Take a look at its location.
From
That's
posterior
ascending.
and lateral.
Sorry, say it differently. What?
From posterior and lateral.
Garrett, what you're saying?
Sensory axons.
Perfect. Sensory axons. You remember, this is the dorsal root. Dorsal root will be here. And this is where the dorsal root ganglion is. And you remember that the axons of the sensory neurons are entering this way to the dorsal horn. So they can synapse with the interneurons located here. So you need to remember that Lissauer's tract basically consists of axons of sensory neurons. It's a bunch of axons of sensory neurons which are entering into the dorsal horn. Does it make sense?
Julia,
Oh,
why is that different? Because isn't what the same thing that the other axons also do?
again?
Why is this mentioned differently? Because isn't that the same thing that the other axons do also?
What do you mean our axons? Which our
Because
axons?
all the axons in the posterior side will be ascending and going to synapse in the gray matter, right?
Yes, yes. But
So
you can see that they're a bit different. The ones which are here are a bit different. So the axons which are meant to synapse with the interneurons in the dorsal horn have to be present in the Lissauer's tract. The axons which aren't synapsing with the interneurons in the dorsal horn, but still entering in this area, could be present in the posterior funiculus. They could be there. So this Lissauer's tract is very special because it contains axons of the sensory neurons, which will synapse with interneurons in the dorsal horn. Okay, so that's why it makes it special. Yeah. Okay, now we're starting the bad thing. It's a bit out of focus, so hopefully you can see what's happening here. So on this picture, we are looking at classification, functional classification of the spinal cords, gray and white matter. So you remember that white matter is organized into the posterior, lateral, and anterior funiculi. Funiculi consist of tracts. There are going to be individual tracts present here. What is another name for tract?
Axon.
It consists of axons, but what is another name for tract? Fasciculus. So funiculi consist of fasciculi. mouthful. Okay, so funiculi consist of fasciculi. That's for the exam. Funiculi consist of tracts. The same thing. Gray matter is also going to be organized, right? We can see that this is the anterior horn. This is the posterior horn. And in some cases, we will also have lateral horn. There's going to be not everywhere, but it is present in the spinal cord where we have additional lateral horn. So the gray matter is also organized, and the organization is called rexed lamina. Okay, so the gray matter is organized into the rexed lamina. Rexed lamina consist of 10 layers. You're going to see that there are going to be 10 layers. Lamina 1 is the most posterior. So lamina 1 is the most posterior. So let's take a look at general organization, right? So we can see that this is lamina number 1, lamina number 2, number 3, number 4. Okay, so 4 is until here approximately according to our university. Okay, so which laminas are present in the dorsal or posterior horn? Lamina 1, lamina 4, all the way. How did we call this connecting area?
Gray commissure.
Very good. Which lamina is present in the gray commissure? Laminar 10. Another way to ask this question, which lamina surrounds the central canal? Because you remember this hole is the central canal, that's where the CSF is present. Lamina number 10. Lamina 10 surrounds the central canal. Which laminas are going to be present in the ventral horn? Lamina number 7, 8, and 9. 7, 8, and 9 are going to be in the ventral horn. So what about 5 and 6? Where they are? They're in the lateral horn. If you have lateral horn, it will be there. So again, so in the dorsal horn, we are going to have laminas number 1 to 4. In the ventral horn or anterior horn, we have laminas 7 to 9. Lamina 10 is around central canal or where the gray commissure is. And in the lateral horn, you have laminas 5 and 6. This is lateral horn. You'll have laminas 5 and 6. Lateral horn is autonomic. Okay, so this is the autonomic area. So this slide is important because now we have to talk about the laminas. Not every lamina is important. The ones that you can see here are the most important. They're the most asked. So let's talk about the first lamina. Lamina number 1. How are they asking you? They're asking you which lamina is the most posterior part of the dorsal horn. So go back. This is the posterior side. This is the anterior side. So the most posterior of the dorsal horn is lamina number 1. Within the lamina number 1, we have a nucleus. You remember that nucleus is a bunch of cell bodies together. This nucleus is called posteromarginal nucleus. This is why the area where this nucleus is located, it is also known as the marginal zone. This nucleus has a very important cells. They're called big Waldear's cells. These big Waldear cells perceive pain. So what is the function of lamina number 1? It perceives pain. Who provides big Waldear cells with the pain information? Lissauer's tract, right? So you remember somewhere here, there's going to be Lissauer's tract. Lissauer's tract, you remember, consists of the axons of the sensory neurons, which are coming in this way to synapse with the interneurons. So if they ask you about big Waldear cell, which kind of neuron is it?
Sensory.
Interneuron.
Interneuron, because where are the sensory neurons?
Dorsal root
In
ganglion.
the dorsal root ganglion. The sensory neurons are in the dorsal root ganglion. So the sensory neuron is going to send the axon, and this axon is going to synapse with the big Waldear's interneuron. Okay, so big Waldear is the interneuron. So pain, lamina number 1. Lamina number 2, where is it located? Ventral or dorsal horn?
Dorsal.
Dorsal.
Because it is right under the lamina number 1, so of course it's going to be in the dorsal horn. In lamina number 2, we have substantia gelatinosa. Okay, that's a substance which is present there. Substantia gelatinosa consists of lots and lots of unmyelinated axons. So in this area where lamina number 2 is present, we have lots and lots of unmyelinated axons. These axons are responsible for the pain and temperature information. So they keep on sending the pain information. I go all the way here. Do I go here? Where did I go? Where did I write it? I didn't write it. Oh, I wrote it. There you go. Rolando's substance. So lamina number 2 contains substantia gelatinosa, but this substantia gelatinosa contains Rolando's substance. Rolando's substance is just a bunch of unmyelinated axons. These unmyelinated axons belong to the sensory neurons. Okay, so this sensory neuron is sitting in the dorsal root ganglion, but it carries information all the way here to the lamina number 2, to the substantia gelatinosa. What's interesting is the following, that these axons are going to be ascending. They go up. The axons in the substantia gelatinosa, they ascend. They ascend through the entire length of the spinal cord. It doesn't matter where you cut spinal cord, you will find substantia gelatinosa there. Where does it eventually end up? It ends up to become spinal nucleus of the trigeminal nerve. There you go. It goes all the way to the medulla oblongata, and all of these axons end up in the spinal nucleus of the trigeminal nerve. We will talk about this nucleus when we talk about medulla oblongata. So is it clear about this lamina number 2? So axons there are unmyelinated. That's why they are gelatinous. That's why they're a bit like having this weird consistency. The axons there are ascending through entire length of the spinal cord, and they end up in the spinal nucleus of the trigeminal nerve, which is located in the medulla oblongata. Okay? Input, we have talked about it, sensory neurons, of course. Take a look, it's not only for the pain, it's also for the temperature. So if lamina number 1 was mostly pain, right? Lamina number 2 is pain and temperature. How is it possible that it senses pain and temperature? Because neurons here use substance P. This is very important. Okay, the neurons here, they use substance P. The axons which are located in this region, or interneurons which are located nearby this region, they are containing high amount of substance P. For the rest of your career, guys, for the rest of your career, substance P, whenever you think about it, P stands for pain. Okay, so substance P. So it's the how do you call it? It's like a neurotransmitter that is responsible for pain. So if you have lots of substance P, you will feel the pain. Okay. Lamina number 3 and 4 contains nucleus proprius. Nucleus proprius, we'll talk about it later, but it has something to do with the cranial nerve number 10. Okay, so you will see that they're connected. This nucleus is connected to the vagus nerve in a way. We'll talk about it later. The interneurons in this nucleus are called antenna cells. Okay, so the interneurons here are called antenna cells. And that brings us to the last lamina, which is very important, highly tested in the neuroanatomy practical and in the neurohisto exam itself. In the lamina number 9, we contain very interesting interneurons. These interneurons are called Renshaw inhibitory interneurons. So to talk about them, let's go for this picture. Okay, so first of all, which lamina is here?
1.
Very good. Which lamina is here?
9.
9. Very good. Which lamina is around central canal?
10.
10. What is sitting in this dorsal horn? What is here?
2, 3, 4 lamina.
Okay, I'll rephrase it. You're right, 2, 3, 4 lamina, but which cells are sitting here?
Interneurons.
Interneurons. Interneurons are occupying this area. Where are the sensory neurons? I asked this question like 50 times already. Where are the sensory neurons?
Dorsal root
Dorsal
ganglion.
root
Root ganglion. They're in the dorsal root ganglion. That's where the sensory neurons are. So what is in the ventral horn?
Motor neurons?
Motor neurons. And these motor neurons are destined to do what? What do they innervate, these motor neurons?
Skeletal muscles?
Very good. They go all the way to the skeletal muscle. So this is somatic innervation, right? It's not autonomic, it's going to be somatic innervation. If it was autonomic innervation, where would the motor neurons be?
Lateral horn.
Perfect. They'll be in the lateral horn. If it was autonomic motor innervation, the neurons will be in the lateral horn. So remember that the entire ventral horn consists of a bunch of motor neurons, somatic motor neurons, which are destined to innervate skeletal muscle. Have we talked about them? How do we call these cells? We talked about them. How do we call them? I gave you a couple of names previous lessons. What is another name for this motor neuron? So this motor neuron here, it's called motor neuron. Yes, our name.
Somatic efferents?
No?
Efferent. What?
Again?
Efferent.
Nope. Efferent is the entire, like you could say efferent, yes, okay, yes, you could, but no, it has another name. Alpha motor neuron. Okay, so alpha motor neuron. That's another name for it. What is another name? Lower motor neuron. You remember we talked about upper and lower motor neurons? I told you that lower motor neuron is going to be here. Another name is lower motor neuron. Where is the upper motor neuron?
Does it come from brain to?
Yes,
Spinal.
exactly. Upper motor neuron comes from brain to the spinal cord. Lower motor neuron goes from the spinal cord to innervate the skeletal muscle, right? There's additional name, anterior horn cell. Anterior horn cell. The same thing. And there's one more. Ventral motor neuron. Okay, I just wrote motor neuron, but you should add here ventral motor neuron because it is in the ventral horn. So these are a bunch of names, and quickly, so you're with me, what are they doing? It's a one cell with all the different names, but what is the cell doing?
Innervating skeletal muscle.
Perfect, it's innervating skeletal muscle. Inside of the lamina number 9, we have Renshaw inhibitory interneuron. Okay, so in the lamina where I am, in lamina number 9, here I have interneuron. And when this motor neuron receives input that it needs to stimulate skeletal muscle, the axon leaves, right, in the ventral root, in the spinal nerve, peripheral nerve, it goes all the way to the skeletal muscle. But this motor neuron gives additional axon, additional one. We call it excitatory collateral, okay? So it gives additional axon, and this axon synapses with Renshaw inhibitory interneuron. So what is the job of the Renshaw inhibitory interneuron? It is always aware of any action potential that motor neuron gives you. So let's simplify it. I am motor neuron, right? My bottle is the Renshaw inhibitory interneuron. Do you see I'm touching the bottle? I have an axon to the Renshaw inhibitory interneuron. What is my other arm? My other arm is another axon. And where is this other axon going?
Muscle.
To the skeletal muscle. Who will tell me that I need to contract the skeletal muscle?
Interneuron.
Interneuron can synapse with me, or brain through the upper motor neuron can synapse with me, right? So that's too much details, but the idea that I want you to understand that I am alpha motor neuron, I am innervating skeletal muscle whenever somebody tells me to do so, right? I will innervate skeletal muscle. But in addition to sending my axon to the skeletal muscle, my other axon is where?
Renshaw inhibitory.
Yes, it's also synapsing with Renshaw inhibitory interneuron. So every time I send an action potential to the skeletal muscle, who else is aware?
The Renshaw.
Renshaw, because you see, I have two branches. Every time I do something with one arm, another one feels it. Every time I do something with one arm, another one feels it. So it doesn't matter whatever your alpha motor neuron is doing to the skeletal muscle, through the excitatory collateral, the Renshaw will feel it. Why do we call it Renshaw inhibitory interneuron? What do you think?
It can inhibit the synapses.
Yes, the Renshaw inhibitory interneuron has very important job. It has a threshold, a certain threshold. If alpha motor neuron starts firing too much action potential towards the skeletal muscle, that means that Renshaw is becoming activated. And if it is activated, it will inhibit the alpha motor neuron. If I inhibit the alpha motor neuron, will there be contraction?
No.
No. So agree with me, every time I'm doing a movement, it's very coordinated, it's very smooth, right? I'm doing everything is great right now. That's thanks to my Renshaw inhibitory interneuron, because it is always kept informed of the function of the alpha motor neuron. It's always kept informed. And you should add something here for your neuroanatomy. There is a certain disease that inhibits Renshaw inhibitory interneurons. If I inhibit Renshaw inhibitory interneuron, what will happen to my muscles?
It's keep contracting.
It keeps contracting. And how do we call it when it keeps contracting?
Tetanus?
Perfect. We call it tetanus. And which disease leads to tetanus? Which bacteria? Which thing that you need to vaccinate your children against? No? It's Clostridium botulinum. Okay, so there is a particular bacteria, the Clostridium botulinum. We call it botulinism. Yeah, that's when, for example, it's in honey or rusty nails. You need to be vaccinated against it. And I think a couple of years ago, have you seen? We have, like in America, there is a rise in tetanus happening. The children are dying because their diaphragms keep contracting. They can't breathe, they die from suffocation. All of this could be avoided by vaccine, but parents choose not to vaccinate their children. They go play on the ground, rusty nails, touch, they get the Clostridium, and as a result, like they die from that. So Clostridium, it loves Rancho inhibitor interneurons. That's the area where it proliferates. It invades these interneurons, as a result, preventing them from doing their job. So that leads to tetanus. Oof, is it clear? Yeah. Okay, this picture is your best friend from now on. Okay, because here we're talking about white matter. White matter possesses tracts. These are the tracts that we need to know, all of them. Okay, so let's try to take a look. How do we call this funiculus?
Posterior.
Posterior funiculus, because it is at the back, right? This is the intraneuron, motor neuron, etc. Posterior funiculus will contain two tracts. There is fasciculus gracilis and there is fasciculus cuneatus. We'll talk about them a bit detail right now. Together, they are making up dorsal column tract. So together, they are known as the dorsal column tract. You have to remember that they are ascending tracts, right? They're light green, so they're ascending tracts. So what is their job?
Send this.
Sensory information.
Sensory. Yes, sensory information all the way to the brain, all the way. Dorsolateral tract or tract of Lissauer is a bidirectional tract. We'll talk about it later. That's where the axons are coming in, but not only coming in, they could also leave this area as well. We'll talk about it later. We have spinocerebellar tracts. We have the posterior spinocerebellar and anterior spinocerebellar tract. Where are we sending information to? Spinocerebellar, so cerebellum. And what do you think is the job of cerebellum?
Control of motor.
Motor, okay. Somebody said it. Oh, go here. You can say this one word.
Proprioception. I forgot how to say.
Perfect. Proprioception. The job of the cerebellum is proprioception. Balance is in the proprioception. We'll talk about it later, but it's in it. Proprioception is your position in space. Yeah, like how are you in space? That's it. So we need to know sensory information about space. So that's why posterior, anterior, spinocerebellar are going to do that. Sixth is the spino-olivary tract. It goes all the way to the olives. Olives are located near the medulla oblongata. We'll talk about them. Fifth, as you can see, there are many of them. Spinothalamic, spino-reticular, spinotectile, and spino-hypothalamic tract. Do you see? They all start with spino and they go up. So all of them must be ascending tract. They must be sensory tracts, right? Fasciculus proprii, we don't talk about it. We know that this is where it is, but actually we don't talk about it, so you can skip it. This eighth as well. Okay, so this one, we don't talk about it as well. Septomarginal fasciculus is important, so it is descending tract, so it is a motor tract. We'll talk about it later. Semilunar or intervascular is going to be mentioned once. So you just have to remember it's called interfascicular because it is between which ones?
The first ones.
Yes, the between gracilis and cuneatus. So between the two ascending tracts, we have one descending, which is intervesicular or semilunar. Now, corticospinal, lateral corticospinal is crucial. It's like they will for sure ask you to show it in the neuroanatomy. So when you do a cross-section of spinal cord, make sure you remember that that's the area where a lateral corticospinal tract is going to be located. Anterior corticospinal tract is going to be located anteriorly, of course. So there are two corticospinal, lateral and anterior. They are the major motor tracts. Anterior corticospinal tract is responsible for innervation of the muscles of your trunk. So skeletal muscles of your trunk. Lateral corticospinal tract is responsible for motor innervation of your limbs. So hands and legs. Which one do you use more?
Limbs.
Limbs. So which tract is more active?
Lateral cortical.
Lateral corticospinal tract, yes. Right next to the lateral corticospinal tract, we have rubrospinal tract. Rubrospinal tract originates in the red nucleus of the brain. We'll talk about it later. That's where it starts. Rubrospinal tract is your major motor tract until you learn to walk. When do you learn to walk?
1 year.
Approximately around 1 year old. So when you turn 1, your major motor tract, the one that allows you to move, is the lateral corticospinal. Until you're 1, it is rubrospinal. So what does the rubrospinal tract allows you to do? What can you do until you're 1? Crawling, very good. Crawling. Rubrospinal tract is responsible not for walking, but for crawling. So this is why animals like cats and dogs, right? Which tract is more dominant in them? Lateral corticospinal or rubrospinal?
The rubro.
Of course, because crawling is identical to how the cats and dogs walk. We crawl the same way. Reticular spinal tract, not so important. Okay, so the 13, 14, you can skip it. Tectospinal is important. We'll talk about it later. Okay, so tectospinal is important. So next class, we're going to have a quiz. It's nothing serious, but I just want to test you from now on just a bit on these tracts, because you will see that in the exam, both neuroanatomy and neurohisto, lots of questions will come from here, lots. Okay, we talked about this. Ascending, descending. You remember the difference between them, right? Ascending are sensory tracts, descending are motor tracts. Yes. How can I name the tract? It depends. If spino is in the beginning, this is a sensory tract because it ascends from the spine up. If spinal is at the end, it is a motor tract because it goes down. There are 3 major sensory tracts. Dorsal column, right? So posterior or dorsal column. You remember it's the gracilis and cuneatus. Gracilis is more medial, cuneatus is more lateral. Spinothalamic and spinocerebellar tracts. Before we continue with the tracts, we need to remember that there are 3 types of neurons. This is very important in neurophysiology as well. We have 3 different types of neurons: first order neuron, second order neuron, and third order neuron. And in some tracts, it's important to know where is the first, where is the second, and where is the third order neuron. First order neuron is going to be your sensory neuron. The sensory neuron that you know, where is its cell body?
Dorsal root ganglion.
Dorsal root ganglion. So you need to remember that sensory neuron is first order neuron. They're identical, just another name. Remember I told you neuroanatomy, lots of names? So there you go. So sensory neuron is first order neuron. Second order neuron is an interneuron. the one that sensory neuron synapses with. Third order neuron is a neuron which transmits the information from the thalamus to the cerebral cortex. So this neuron is in the brain. Okay, so intraneuron will go all the way to the thalamus and will synapse with the intraneuron there. So it's also intraneuron, but its location is between thalamus and brain, cerebral cortex, sorry. Okay, everything else is not important. This is important, medial lateral rule. So this is, for some reason, gets confusing. Okay, so medial lateral rule. So sensory neurons that enter at the low level of the spinal cord are always going to be more medial. Why? Because they have a longer distance to go. Think about it this way, right? So if this is my spinal cord right here, and this is the brain, what is my destination? Where am I going? I'm going towards the brain. So if my sensory neurons entered into the spinal cord very low, right? Very low, very far away from the brain. Should they keep medially or laterally? They should keep medially because they have longer distance to travel. It's very identical to highway. If you need to take an exit, you keep right, you keep lateral. But if you don't need to take the exit, you keep left, you keep medial, you don't go towards the exit. So if you are from the lower part of the body, you are located medially in the spinal cord. But what if I'm from the upper part of the body? I just entered, right? And soon I'm going to take another exit. I keep right, I keep lateral. So you remember we have said that we have the gracilis and we have the cuneatus, the two tracts, right? They're the sensory tracts. Gracilis carries information from where? From upper limb or from lower limb?
Lower limb.
Yes, because it is located
Medially.
medially. Cuneatus, does it carry information from lower limbs or from upper limbs?
Upper limb.
From upper limb because it is located
Laterally.
laterally. So do you understand this medial lateral rule? The more lower you start, right, at the spinal cord, the more medial you'll be in the spinal cord. So what is the level, right? So how do we know which level is the low, which level is the high? T6. Any axons which are entering inferiorly to the T6, they are from the lower limb, so they are going to be the part of the gracilis, so medial. Any axons entering above the T6, they're from the upper limb, so cuneatus laterally. Okay, the same picture for you, very clean, so you studied, yeah? So you make sure that you studied. Spinothalamic tract. Spinothalamic tract is going to be going from the spinal cord all the way to the thalamus and then to the cerebrum. It doesn't stop at the thalamus, it goes to the cerebrum. What is its job? Pain and temperature. Spinocerebellar, proprioception. That's the word that you need to always remember. As soon as you see cerebellum, that's the word. We'll talk about it later. This is additional information for you, but I strongly suggest you guys study this on your own. It's very nicely visible here. So it's repetition, okay? So it's always repetition. White, gray matter, dorsal root, ventral root, all the different fasciculi, all the most important tracts. Make sure that you see that these are your sensory tracts, right? So all of them here are going to be your sensory tracts. This is general information about the sensory tracts. Which kind of things are they carrying? Pain, temperature, proprioception, light, touch. Here you can see this picture. You can see this particular pattern, right? So you can see the pattern. This is called crossing. What you see here, this is known as the crossing. So if my axons are entering from the left side, they don't necessarily stay on the left side. There are areas where they start to cross to the other side. So let's imagine that this is the left side, this is the right side. And I tell you that my right spinothalamic tract above T6 region is damaged. My right spinothalamic tract above T6 is damaged. What I will not be able to sense? Pain and temperature. But where I will not be able to sense?
Left side.
Left side, very good, because it carries information from the left side. Do you see? And I said above T6, so my left.
Below T6.
So
Then
above,
upper limbs.
my left what?
Lower limb.
My left upper limb.
Upper limb.
Because above T6, this is going to be the upper limb. So just because it says here like this, it doesn't mean that this is the T6 itself. So it's my mistake. I did it wrong. I should have said that this is the T6. I apologize. Okay, so this is the T6. My mistakes fully. Okay, so this is the T6 right here. So the crossing has happened here. So it will be arm. Do you get it? Because it's above the T6. It's my mistake. So how do we call this when it goes to the opposite side?
Crossing.
Crossing. But how do we, what's another word that we can use here? The other word?
Yes.
Desiccation?
We just say contralateral. The contralateral means that it's crossing. Ipsilateral means that it's not crossing. So take a look at these tracts, right? Take a look at these tracts. Which sensation is always contralateral? Which sensations are always contralateral? So pain and temperature, do you see it crosses? Contralateral. Conscious proprioception is always contralateral, right? Light touch, do you see? It switches, it crosses, contralateral. Motor is always contralateral. Which one is the only one which is ipsilateral? It stays on the same side. Unconscious proprioception. Who deals with unconscious proprioception? The cerebellum. So let's do this again so you get it, guys. Tracts do not stay on the same side, right? They don't. The sensory neurons, when they go into the spinal cord, they never stay on the same side. They always cross. And in neuroanatomy, it's important to know where they cross. It's very important because we have to know where the crossing happens. We'll talk about it later, but for now, I just want you to understand that the crossing actually happens. So if the crossing happens, the neurons which are carrying the information from the left side are creating a tract which is located on the right side. So if I damage this tract, if I have a stroke, I have infection, damaged, I cut myself somehow, I cut my right side completely, the tract, I lose the sensation on the left side. This is the contralateral component, right? So if your patient comes into you and says, I cannot feel pain in my right arm, what do you start thinking? He can't feel pain in the right arm. What do you start thinking?
The left side, but above T6.
Yes, but which tract pain?
Spinothalamic
Tract, right? So that if I can't feel pain in my right arm, that means that my spinothalamic tract on the left side above T6 is damaged. Something is compressing that part. Do you get it? The symptom says the right hand, but you don't care about the right hand. You're thinking what is inside, right? It's crossing, so that means it's going to be on the left side. And notice that it's not only for sensory, it's also for motor. If a patient comes into you with a stroke and says, I cannot lift my right arm, I cannot lift it, where is the damage?
Corticospinal tract.
But on which side?
Left side.
Left
On the
side.
left side. The damage is on the left side. Do you get it? The only thing that stays ipsilateral is the one that involves cerebellum. That's the only thing. Okay, so anything with cerebellum, it will stay on the same side. That's it. So if your patient says, okay, what can he say? Cerebellum. If your patient says that, I don't know, like I am tilting towards the right side, I cannot stand straight, I am tilting towards the right side, where is the damage?
Left side.
Right.
Right side.
Spinal
So
thalamic.
this is cerebellum, it's your balance. I'm tilting towards the right side. That means the damage is on the right side because I am tilting towards it, right? So there is some kind of damage there. Yes? Okay. This is for you guys. You're gonna read it at home. Okay, so this is for you. It's nothing interesting. Well, it is interesting. It's just we're talking about that it is contralateral. Okay, so everything is contralateral. Okay, the same is here. You're just gonna read it at home. It's everything that we have talked about. Don't forget, spinocerebellar is the only one which stays ipsilateral. Okay, so cerebellar are always ipsilateral. Anything with the brain are always contralateral, always. Okay, so remember that. The same idea, just more details. Okay, so this is, you're gonna read it at home. There's nothing difficult, it's just additional information in terms of like structures. Okay, so you can see that this is anterior spinocerebellar tract. Do you see that it stays on the same side? Eventually, it stays on the same side. It has crossed, but it has crossed twice. So it ended up on the same side. It's ipsilateral, right? For dorsal, it's the same, right? So it's going to be ipsilateral. So again, just additional information, but the most important things we have covered. Let's take a look at this part. So tract of Lissauer, so these are additional sensory tracts that we're talking about. Tract of Lissauer, we have talked about it. It possesses axons of the sensory neurons. These axons are going to be synapsing with the second order neurons that are located in the substance of Rolando, in the substantia gelatinosa or laminar number 2. Nucleus proprius, as you remember, was lamina number 3-4. Okay, so this is where it synapses. That's the most important part there. Once they have synapsed with those things, you're going to see that they are going to cross. They're going to go on the contralateral side, creating spinothalamic tract. So you remember spinothalamic tract? Where does it originate from? From the tract of Lissauer. So tract of Lissauer comes in into the dorsal horn, cross to the other side, creating spinothalamic tract. Spino-olivary tract has another name. It is also known as the Helwig's tract. It is going to be located in the anterior funiculus of the spinal cord, and it is responsible for the unconscious proprioception. Unconscious proprioception, it basically means that you don't have to think which joints to flex when you walk. You don't have to think about it. You just go, okay? You're just walking. Everything is done for you by the spino-olivary tract and cerebellum, okay? So spinocerebellar tracts are going to be there as well. So that's the most important thing. So remember that this information is going to the olivary bodies. These olivary bodies are located in the medulla oblongata. And the information is from your muscles and tendons and skin. So just your general position where you are, right? So if I'm sitting right now and I want to get up, I don't need to think that I need to contract my flexors of the legs, right? So I don't need to do so. So it's done for you. That's the unconscious proprioception, thanks to the spinal olivary tract. Spinotectal is actually very interesting because it inhibits or controls pain sensation. Spinotectal tract is the tract which is responsible for your pain tolerance. How well do you handle pain? So everybody has their own pain tolerance, right? So there are some people that feel almost no pain when they cut themselves or something like this, right? Some people, you scratch them a little bit and they say it hurts. That's the fault of the spinotectal tract. The threshold of pain is set by this tract. Spinal-reticular tract is responsible for automatic responses to pain. What is automatic response to pain? If I hurt you right now, what do you do?
Reflex.
Reflex. So what is the reflex? You do this, right? So you remove your limbs or whatever I'm hurting from the cause. Whose job is it? Spinal reticular tract. It's responsible for you saying, ouch, it hurts, and doing this. Okay, so that's what spinal reticular tract does. Motor tracts, so the descending tracts, we have two types of them, the somatic and the autonomic, you remember, right? So somatic is the voluntary, autonomic is the non-voluntary. We're going to talk about the corticospinal tracts and the subconscious tracts. The corticospinal tracts, there are three of them. We have corticobulbar, lateral corticospinal, and anterior corticospinal. We kind of talked about them, right? Anterior corticospinal is responsible for the contraction of what? Of which skeletal muscles? Where? Of the trunk. And lateral corticospinal is of the limbs. So which one do we use more? Of course, the lateral one. Corticobulbar tract controls your face. So skeletal muscles of the face, okay? I'm talking right now, I can raise my eyebrows, I can do this, I can smile, okay? So this is all voluntary and it is done by the corticobulbar tract specifically, okay? So all of it is voluntary. Okay, we talked about this part before. You need to remember that where do corticospinal tracts cross? It's very important to remember where do they cross. Approximately at the same level, we call that level medial lemniscus. Medial lemniscus is a junction between spinal cord and brainstem. It's the area where medulla oblongata and spinal cord meet up. Okay, so that's what's medial lemniscus. Lower motor neuron is which one? What's another name for it?
Alpha motor neuron.
Very good. Alpha motor neuron, where is it located?
In anterior.
Very good. Anterior horn of the spinal cord. Lower motor neuron can also be known as the second order motor neuron. Upper motor neurons are sitting in the brain. They have another name. They're also known as the Betz cells. Betz cells are also known as the first order motor neurons. If you have damage to upper motor neuron or lower motor neuron, what you're going to see? Flaccid or spastic paralysis. Because think about it, if lower motor neuron is damaged, what is the job of the lower motor neuron?
To move the skeletal muscle.
Exactly, it innervates skeletal muscle. So if I damage that neuron, can skeletal muscle contract?
No.
Exactly, so there will be nothing. So you're going to see that there's a flaccid paralysis, zero movement. If you damage the upper motor neuron, right, the lower motor neuron can still fire the action potential to the muscle. So you will see that there's going to be spastic. There's no significant atrophy. The muscle still receives the input from the lower motor neuron. But if the lower motor neuron is damaged, there is definitely atrophy. Fasciculations and fibrillations are generally the how do you call it? It's generally like the muscle itself contracting. Okay, so the muscle itself just involuntary doing this particular contraction. Typically, lower motor neuron can inhibit it. So involuntary impulse of the skeletal muscle can be inhibited by the lower motor neuron. So if you remove lower motor neuron, the skeletal muscle is free to do whatever it wants to do. So fasciculations and fibrillations are present. But here they're not going to be present because, I repeat, in this defect, lower motor neuron is intact. It's still there, so you can still inhibit the skeletal muscle. Hyperreflexia, hyporeflexia, because lower motor neuron participates in the reflex. Remember? Sensory neuron, interneuron, lower motor neuron, or alpha motor neuron. So if you damage the lower motor neuron, you will see that all of the reflexes are very diminished, almost non-existent. But if you damage the upper motor neuron, right, you still keep the lower motor neuron intact, so there is going to be a reflex. Babinski, we'll talk about it next lesson. Okay, I'll show you the video, or you can do it at home. Just research what is Babinski reflex. Babinski reflex is present in babies. We adults no longer have it. If you're a healthy adult with no brainstem damage, you will see that you should not have Babinski. So if it says that Babinski reflex is present, you have brainstem damage. That means that your upper motor neuron is damaged. What is Babinski reflex? Imagine if this is my sole, yeah, the sole of my foot. This is the sole of my foot. You take a sharp object and you do this movement on your foot. Okay, so this movement like this on your foot. Babinski reflex in babies will do like this. Okay, so the toes will go this way. If I say that Babinski reflex is present, that means that you, adult, have the same reflex as a baby. Your brainstem is damaged. Your toes do this. Babinski reflex not present is when your toes do this. Okay, if I do this movement, they do this. So they do flexion. Okay, this is flexion. This is if it is not present. If it is present, toes do extension. You get it? But watch a video at home. Okay. So here we're just the same idea. We have to understand that there is something which is called pyramidal tract. We have pyramidal tracts and we have extra pyramidal tracts. Corticospinal tracts, so your anterior and lateral corticospinal tracts, together with corticobulbar, are considered to be pyramidal. Pyramidal tract are voluntary. They're the ones that, for skeletal muscle, voluntary. Why do we call them pyramidal? Because they form two pyramids on either side of the medulla. The way they pass, it's like they're forming two pyramids. So that's why they call it pyramidal tracts. Extrapyramidal tracts are not forming pyramids. They're above the pyramids. They're the involuntary muscle situation. So whenever you actually hear the word pyramidal tract, always think voluntary. Which tracts are voluntary? Corticospinal and corticobulbar. Extrapyramidal tracts are involuntary. which ones are involuntary. We'll talk about it in a sec. So this is more detail about lateral corticospinal, read it at home. More details about anterior corticospinal, read it at home. And corticobulbar, read it at home. So now the involuntary, subconscious, another name, extra pyramidal tracts. Okay, that's another name for them. There are 4. Vestibular spinal, tectospinal, reticular spinal, and rubrospinal tract. Vestibular spinal tract is responsible for your posture. Okay, so limbs, it affects your limbs, like whenever you're, I don't know, like trying to find a balance, right? If you stand on one leg, trying to find the balance, your limbs are moving, trying to balance your body. That's your vestibular spinal tract. Which part of your brain, which part of your body senses the balance? The inner ear. We'll talk about it this semester as well. In the inner ear, we have semicircular canals, we have vestibule, we have saccule, macule, these structures, and they're responsible for you realizing where you are, right? Tectospinal tracts are responsible for this. So if I clap right now, all of you should have, you know, blinked like that. That's the tectospinal tract, because it's your reaction to loud noises, your reaction to bright color, your reaction to like all of this sudden movements, right? So you always do these jerky movements. Your body always kind of tries to protect itself, right? So that's the tectospinal tract. It's involuntary, right? It's fully involuntary. You cannot control it. Tectospinal tracts receive information from 2 areas, from the superior colliculus and inferior colliculus. We will talk about them in the anatomy a bit. Superior colliculi are responsible for visual information. Inferior colliculi are for the auditory. So sometimes you have a patient, right? And he hears everything you say. Don't get me wrong, like he hears, you have a conversation with him, he fully hears you, everything is great. But then you do this right next to his ear and 0 reaction. No movement like that whatsoever. So typically we expect that there is something wrong with the tectospinal tract, specifically the one which was receiving information from inferior colliculus, right? Because it is auditory. The same idea for visual. You have some patients, they see you, everything is great, but you clap in front of their eyes, right? And 0 reaction, they don't blink, they're not trying to protect anything. That means that superior colliculus was damaged somehow. Reticular spinal tract is responsible for your respiratory muscles. So you don't control your diaphragm whatsoever. I mean, you can. I can take a deep breath and I can exhale, but still, on a daily basis right now, I'm not controlling my diaphragm to contract. So that's reticular spinal tract. The rubrospinal tract sends information to flexors and extensors muscle. So this tract is the one which is also for crawling. You remember, rubrospinal is for crawling, but it's also just in you particularly, because you're not a baby, any information to flexor or extensor. So I don't know, you're falling and suddenly you need to make sure that you aren't falling, right? You're grabbing the table not to fall. That won't be lateral corticospinal, because lateral corticospinal is voluntary. It will be a rubrospinal tract telling your flexors and extensors of the arm to stretch and grab the table as you're falling, because it's unconscious. It's your body trying to maintain its position. Yeah. Okay, this picture is for you, so you can start getting used to it. So spend some time on this picture. These are all the nuclei that are present in the brain, right? So we have the basal ganglia, we have thalamus. Take a look at the colliculi, right? Superior, inferior. You can see they're right next to the tectal area, red nucleus, pons, vestibular, cerebellum. So get used to this picture, because we will talk about them later. Ascending tracts, descending tracts, you got the difference, right? Sensory, motor. Quickly give me an example of ascending tracts.
The dorsal column.
Very good. Dorsal column tract, the fasciculus gracilis, fasciculus cuneatus. Yeah. Anything else? Spinocerebellar and spinothalamic, right? So these are the three major sensory. Give me a couple of descending tracts.
Tectospinal.
Very good. Tectospinal. Is it pyramidal or is it extrapyramidal?
Extrapyramidal.
Very good. Tectospinal is extrapyramidal because it's involuntary. Give me an example of descending pyramidal.
Corticospinal.
Perfect. Lateral anterior corticospinal tracts together with corticobulbar, right? Okay, guys, that's it for today. This is enough for today. So try to study spinal cord so I can ask you on this next class. Okay, so what we do the slides so you have some information. So try to study it, yes? So it was a lot. Thank you so much